Hi everyone, my name is Srishreya, and today I'll be presenting about the genetic and age-related factors in Alzheimer's disease progression. A little bit about me before we get started. I was born in California, and that's currently where I'm presenting from. I go to American High School. I'm currently a senior, and I love learning about new medical discoveries, and I'm very passionate about neuroscience. So, without further ado, let's get into it. So, first, I'll be talking about the introduction to Alzheimer's disease. Alzheimer's disease is a prevalent neurodegenerative disorder which primarily affects those in the ages of 65 and older. However, it can also affect those in the age range of 30 to 45 years old. Approximately 1 in 9 people are diagnosed with Alzheimer's disease, which accounts for 6 million Americans affected. It's characterized by erratic amyloid deposits, such as cerebral amyloid and amyloid plaques, which are also the presence of neurofibrillary tangles and neuronal death. This Alzheimer's disease is the leading cause of dementia, impairing memory, cognition, and behavior. Typically, 4 to 8 years post diagnosis, people are able to survive. However, some can also go up to 20 years depending on various factors, including genetics or their environment. The stages of Alzheimer's disease. So it starts out at an early age. This can show signs of memory loss, such as forgetting specific places or objects. And it can also show like subtle changes in their cognitive abilities. However, they're still able to function independently. During their middle stage, this is the longest stage and lasts many years. However, there are more noticeable symptoms such as word confusion, mood swings, or cognitive impairment. In their later stages, there's a severe cognitive decline and a loss of ability to respond to their environment or stimulus, and they require full-time care. The key progression factors, such as the symptoms worsening or their language, reasoning, and ability to perform or their behavior, and it impacts their ability for their daily tasks and their independence. So on the picture on the right, we have a healthy brain. You can see the cerebral cortex is intact as well as the hippocampus. However, in the mild Alzheimer's disease, we can see a shortage in the cortex and also a shrinking of the hippocampus. And in their very severe or later stages, we can see a severe shrinking in their hippocampus and their cortical shrinkage. Their hippocampus is mainly accountable for their moods or their speech. So the shrinkage is very crucial. Next is the study objectives accounting with my paper. So in my study, I wanted to explore the interactions between APOE variants and Alzheimer's disease progression and investigate the age-related trends in Alzheimer's diagnosis, then analyze cognitive impairment across those different diagnostic groups, and evaluate the data and examine the key diagnostic variables, which is the Alzheimer's disease diagnosis, probable Alzheimer's disease, possible Alzheimer's disease, and the age at the initial visit. With that information, I conducted a statistical analysis to compare the age distribution and their cognitive scores and gain insights into the genetic and environmental factors that influences their Alzheimer's disease. Next is the APOE gene in Alzheimer's disease. So the alipoprotein E encodes the protein involved in lipid transportation, metabolism, and especially in the brain. It has 3 main alleles, APOE2, 3, and 4. In my research paper, I focused on alipoprotein 4, which has a higher risk and earlier onset of Alzheimer's disease. However, it is also notable that Alzheimer's has a gene APOE2, which potentially protects it. The research implications include its ability to improve preventative strategies and treatments, and it's very critical for understanding the complexity and the heterogeneity of this disease. Moving on to my study population and data source. The data source I used was the Alzheimer's Disease and Research Center's program. This had 185,832 individuals, um, and the participants spanned from like a spectrum of no cognitive impairment to dementia. The variables I used are stated here. And this is a longitudinal clinical data collection that started in 2005. And these are referral based or volunteer case series, and it's not statistically representative of the US population alone. Talking about my diagnostic variables. The first one is the standardized classification of Alzheimer's disease diagnosis. This is based on the initial clinical findings when they first come in and are diagnosed with Alzheimer's disease. The next is the probable Alzheimer's disease. This is supported by neuroimaging and the absence of significant comorbid conditions. The next is the possible Alzheimer's disease. This is an atypical feature or any other contributing factors that leads them for the diagnosis or it complicates it further. And I also included the age at the initial visit as a continuous variable used to examine the age-related trends. This significance helps categorize participants based on the diagnostic evidence and allows for analysis of age-related patterns in Alzheimer's disease. So when I conducted the statistical analysis, I conducted a two-sample t-test. The purpose of this t-test was to compare the mean scores and the age distribution between the groups. I used a significance level of 0.05, and these comparisons were all pairwise comparisons between all the diagnostic groups. My results concluded in the p-value of 0 for all the comparisons, stating that there was a significant difference in mean scores between the groups. This evidence helps against the null hypothesis of equal means, and it highlights the significant disparities between the diagnostic groups. This overall underscores the role of age and cognitive function in Alzheimer's diagnosis and suggests a further need of research into the underlying factors for Alzheimer's disease. So this is an image of my sample of t-tests. I'll just give you a minute to look over it. Here we can see the mean, the variance, and then the p-values are all zeros. Moving on to my key findings. I found that there is a significant difference in the age distribution and cognitive scores across all the diagnostic groups. The p-value of 0 for all the pairwise comparisons indicated that there was a strong statistical difference, and there was a direct correlation between probable Alzheimer's disease and those diagnosed with Alzheimer's disease. The age at the initial visit plays a crucial role in the Alzheimer's diagnosis and the progression. The mean age for this was about 71 years, and there are complex interactions observed between the genetic factors and the progression. This highlights the heterogeneity of Alzheimer's disease across the different diagnostic categories and suggests that there is a further need of investigation into the genetic and environmental factors and its influence. Next is brain regions affected by Alzheimer's disease. The main brain regions affected by this disease is the entorhinal cortex and the hippocampus. These areas are very crucial for one's language, ability to reason, and their behavior and outcomes to different stimuli or their environment. There's a progressive destruction of nerve cells, and its impact is mainly on one's memory, their cognitive function, and their daily task performance. There are amyloid deposits and plaques, which are crucial to the brain, and neurofibrillary tangles, and much of there is neuronal death. So on the right, you can see the green portion right here is entorhinal cortex, and over here is the hippocampus. Next is the APOE4 structure. So this is one of the main 3 alleles of APOE gene. This encodes in protein and is involved in lipid transportation and metabolism. It's important for the brain function, and it's associated with a higher risk and earlier onset of Alzheimer's disease. This differs from APOE2 and 3 based on its neurobiology, and its structures affect the role in lipid metabolism and AD pathology. So the protein structure here is an overall shape and is showing like a ribbon-like structure with different colors representing different amino acid residues. Moving on to my conclusions. I concluded that there was a significant difference in age distribution and cognitive scores across the different diagnostic groups. The complex interactions between these genetic factors in AD progression and their age at the initial visit played a crucial role in the AD diagnosis and progression. The APOE4 associated with the higher risk and earlier onset of AD, and APOE2, which is potentially protective against it. There is a need for personalized treatment and approaches based on these genetic profiles, and it also underscores the complexity of AD's underlying mechanisms, and it highlights the importance of early diagnosis intervention. Lastly, for future directions, there is a need for further investigation into genetic and environmental factors that influence AD and enhance earlier diagnostic techniques. The study role of APOE in lipid transportation and metabolism in the brain, and for improving understanding of AD's complexity and heterogeneity, and focus on preventative strategies for higher risk individuals. Thank you.
Awesome. Thank you so much, Srishreya. This is a very interesting presentation. I'm curious, you mentioned that there were many types of environmental factors to look into next. Do you have any guesses as to what type of environmental factors you would look into?
A few environmental factors would be like their social circle or their ability for their communication, how they talk to other people, the people that they're surrounded by can also like alter their Alzheimer's disease diagnosis.
Gotcha. And these are, I assume you would look into this for also like the older population. Is there anything like you mentioned that there are some signals at like a young age? When you say young, what do you mean by young?
Young age typically means like 30 to 45. This is not very common to be diagnosed with Alzheimer's disease at the ages of 30 to 45. However, there are some cases that have been found.
Gotcha. Are there any variables that you wish you had access to when running these tests?
A few variables I wish I had access to would be the age. Though I had the age at the initial visit, I would have liked to see the age of their progression based on like their late stage, how old were they, or their earlier stages, how old were they. So that would have
Gotcha.
been more interesting to see.
Yeah, like the progression of age and yeah, cool. And then finally, what was the most unexpected thing that you learned during your research process?
Probably the genes related to Alzheimer's disease, learning about APOE, which is polyprotein E, learning that though there are 3 variations of that, one of the variations, which is APOE2, potentially protects people against Alzheimer's disease. So I think that was a very interesting discovery.
Yeah, very interesting. I have many relatives in my family that have Alzheimer's, so this was very pertinent. So thank you for looking into this and appreciate your presentation.